6 miles below the surface of the Pacific Ocean, where the pressure is over 1,000 times what you feel right now, something alive is eating. Scientists just found it. Thousands of worms and clams thriving in total darkness, feeding on poison gases seeping from the Earth's crust. We are going to travel down there together, past the ghostly snailfish with bones that never hardened, past creatures eating plastic that you threw away, all the way to a place that rewrites what life is allowed to do. If you enjoy this journey, like and subscribe. We go deeper every week. Brace yourself. We begin. On March 23, 1875, a British ship called HMS Challenger stopped in the middle of the Pacific Ocean and dropped a rope into the water. This was how scientists measured ocean depth back then. You tied a weight to a rope, dropped it over the side and counted how long it took to hit bottom. Simple. They had done it hundreds of times on this voyage already, but this time the rope kept falling. The crew watched as it unspooled off the reel, past the depth of any ocean they had measured before, past where it should have stopped. The weight kept pulling the line down into blackness so deep that the rope ran out before it hit anything. They added more rope and waited. When it finally stopped, the measurement came back at over 4.5 miles down. The scientists aboard looked at each other, baffled. They had just found a hole in the ocean floor so deep it broke their equipment. Nobody had expected this. The scientific world in 1875 assumed the ocean floor was mostly flat, a gradual slope from the coastlines into one giant level basin. Simple, boring, done. This one sounding destroyed that idea completely. The trench they discovered would eventually be named the Mariana Trench, after the nearby Mariana Islands. Its deepest point, later called Challenger Deep, after the very ship that found it, would eventually be measured at roughly 7 miles below the surface. That is deeper than Mount Everest is tall. You could drop the entire Himalayan mountain range into this trench and still have room left over at the top. Here is the number that makes it real. The average depth of the world's oceans is about 2.5 miles. The Mariana Trench goes nearly 3 times deeper than that. It is an abyss inside an abyss. The scientists aboard HMS Challenger had no idea what lived down there, or whether anything lived down there at all. The leading scientific opinion of the time was that deep ocean water below a certain depth was simply dead. Too cold, too dark, too far from the sun. The idea had a name, the abiotic zone, the zone where life ends. They were about to find out how wrong that was, but finding it out would take over a century, because getting 6 miles down is not like diving into a pool. The pressure at that depth is so extreme that most submarines ever built would be crushed like a soda can before they got halfway there. The crew of HMS Challenger recorded their discovery, packed up their ropes, and sailed on. They had no way to go deeper. They had no way to see what was down there. All they could do was mark the spot on a map and leave it as a question for the future. That question sat unanswered for 85 years, and when scientists finally got the technology to go down and look, what they found at the bottom made the original discovery seem almost ordinary. Because the hole itself was not the strange part. The strange part was what was living inside it, what it was eating, what it was made of, and what it was doing down there in the dark, completely cut off from the rest of life on Earth for millions of years. The deeper we go into this story, the weirder it gets. Most people hear 6 miles and think of a long jog, a bike ride, the distance between 2 suburbs. Down is different. 6 miles of ocean stacked above you is not a distance. It is a weight. It is a crushing, suffocating column of water that pushes on every surface of every object with a force that the human body cannot begin to process. Before we go any further into what lives down there, you need to feel how far down it actually is. Picture the Empire State Building. It stands 1,450 feet tall. Now stack 36 of those on top of each other. That is how deep the bottom of the Mariana Trench sits below the surface of the ocean. Picture Mount Everest, the tallest mountain on land, 29,000 feet of rock and ice. Drop it into the Mariana Trench. Its summit would sit over a mile below the waves. You could walk over it in a submarine and not know it was there. Picture a school bus falling straight down through water at a constant speed. It would take over an hour to reach the bottom. That is how far down we're talking. And here is the thing about those 6 miles. They are not empty space. Every inch of that distance is filled with water pressing harder and harder the deeper you go. For every 33 feet you descend, the pressure on your body increases by the weight of the entire atmosphere above you. By the time you reach the bottom of the Mariana Trench, the pressure is over 16,000 pounds per square inch. That is like having 50 jumbo jets balanced directly on top of you. That pressure is why exploring this place took so long. Steel warships that can sail through hurricanes would crumple on the way down. Most materials humans have ever made simply fail under that kind of force. Building something that can survive it requires an entirely different way of thinking about engineering. And here is what makes it stranger. The deepest military submarines ever built can reach about 1.5 miles down. The Mariana Trench goes nearly 5 times deeper than that. We sent a man to the moon before we sent anyone to the bottom of our own ocean. The trench is also long. It stretches 1,580 miles across the Western Pacific, roughly the distance from New York City to Denver. It is a scar on the ocean floor so vast that you could fit the entire state of Texas along its length and still have miles to spare. This is the environment where the creatures we're about to meet were born, where they evolved, where they have spent their entire existence for millions of years completely surrounded by crushing darkness. And they are not struggling down there. They are thriving. The fish, the worms, the microbes, they do not experience the pressure as something to survive. For them, it is simply home, normal, the only world they have ever known. Which raises a question that gets more interesting the longer you sit with it. If life can build a thriving ecosystem at the very bottom of the ocean, under conditions that kill everything humans have ever made, what exactly are the rules of biology? Who wrote them? And how many of them are wrong? The answer starts with the rocks. The Mariana Trench was not always there. About 50 million years ago, 2 of the Earth's massive tectonic plates began a slow-motion collision that is still happening right now, today, under the Pacific Ocean. On one side, the Pacific Plate, an enormous slab of oceanic crust the size of a continent. On the other, the Mariana Plate, smaller but stubborn. When 2 tectonic plates meet, and one is heavier, physics takes over. The heavier plate dives under the lighter one in a process called subduction. The Pacific Plate lost. It began sinking beneath the Mariana Plate at a rate of roughly 2 inches per year, grinding downward at an angle into the Earth's interior. As it sank, it dragged the ocean floor down with it, carving a deep groove into the crust. That groove is the Mariana Trench. 2 inches per year sounds slow, but over 50 million years, it adds up to a scar nearly 7 miles deep, and it is still going. Right now, as you listen to this, the Pacific Plate is sliding under the Mariana Plate, pulling the trench floor slightly deeper. The bottom of the ocean is slowly sinking. The grinding of these 2 plates also creates enormous heat and stress in the crust below. Gases get squeezed out of the rock. Methane, hydrogen sulfide, and other chemicals seep up through cracks in the trench floor, bubbling into the cold water above. Those chemical seeps are not just geologically interesting, they turned out to be the foundation of an entire food chain. But there is another consequence of this collision that most people never think about. When a tectonic plate subducts, it does not disappear cleanly. It bends, it cracks, it releases energy. The Mariana region is one of the most seismically active zones on Earth. Earthquakes happen there regularly, sometimes powerful enough to send ripples across the entire Pacific. The trench itself sits in a region surrounded by volcanic island chains. The same tectonic process that carved the trench also fuels underwater volcanoes nearby. The Mariana Arc, a curved chain of volcanic seamounts, runs parallel to the trench just to the west. Some of those volcanoes are active, pushing lava out of the seafloor, while 6 miles away, In the trench, the bottom sits in frozen stillness. This is the geological context for everything that lives there. The trench is not a passive hole. It is an active wound in the Earth's crust, constantly being pulled deeper, constantly releasing chemical energy from below. That chemical energy is not wasted. Life found a way to eat it. Before scientists understood chemosynthesis, the trench seemed like a dead end, a place where particles slowly drifted down from above and rot, a place where nothing could really establish itself, a cold, dark, pressurized desert. Then they looked closer at the seeps, and what they found there made the entire previous picture collapse. The trench is not a graveyard, it is a generator. But to understand what powers it, we first need to understand what the pressure actually does to living things. Because before anything else, any creature that wants to survive 6 miles down has to solve the pressure problem, and the solution evolution came up with is stranger than anything an engineer would design. Take an empty styrofoam cup, seal it in a bag and drop it to the bottom of the Mariana Trench. When you pull it back up, it will be the size of a thimble. The pressure will have compressed every air bubble in the foam until the cup is a dense, tiny nugget of its former self. Scientists actually do this as a demonstration. They send styrofoam cups down and bring them back as souvenirs. Now imagine that happening to a lung. At the bottom of the Mariana Trench, the pressure is over 16,000 pounds per square inch. At sea level, you're under about 14 pounds per square inch of air pressure. The difference is over 1,000 times. Every square inch of your body, right now, has about 14 pounds pressing on it from the atmosphere. At the bottom of the Mariana Trench, that number becomes 16,000. This is not a problem your body can adapt to. It is not a matter of training or preparation. The air pockets inside you, lungs, sinuses, inner ear, would collapse instantly. The nitrogen dissolved in your blood would behave in ways it was never designed to. Your cells would rupture. The process would take seconds, yet the creatures living there show no sign of distress. The reason is fundamental. They do not have air inside them. Any creature that evolved in the deep ocean long ago lost or never developed gas-filled cavities. Fish near the surface use a swim bladder, a gas-filled sac, to control buoyancy. The snailfish at the bottom of the Mariana Trench has no swim bladder at all. Its body is mostly water, gelatinous and soft. And because water does not compress the way air does, the pressure inside its tissues matches the pressure outside. It is in balance. The same logic applies to the bacteria, the worms, and the single-celled organisms down there. They are mostly liquid. Pressure acts on them differently than it would on anything with air pockets. But high pressure still causes damage. It distorts the shape of proteins, the molecular machines that run every biological process in every living cell. When proteins lose their shape, they stop working. At high enough pressure, this kills a cell just as surely as fire or poison. So how do creatures at the bottom solve this? They evolved a chemical that fights back against pressure directly at the molecular level, a stabilizer built into their cells. The name of that molecule is almost impossible to say in casual conversation, but its effects are extraordinary. And when scientists finally figured out what it was and how it worked, it opened a question nobody had thought to ask. If life invented a molecule to fight pressure, what else has it invented that we have not found yet? On January 23rd, 1960, 2 men climbed into a steel sphere about the size of a walk-in closet and began sinking toward the bottom of the Earth. Their names were Jacques Piccard and Don Walsh. Piccard was a Swiss oceanographer. Walsh was a United States Navy lieutenant. The vessel they were riding was called the Trieste, a design built by Piccard's own father, a man who had already set the world record for the highest balloon flight. The Piccard family, in other words, made a habit of going to places no human had ever been. The Trieste was not sleek. It looked more like a floating gas tank with a tiny sphere bolted to the bottom. The upper section was filled with gasoline, not for fuel but for buoyancy, because gasoline is lighter than water. The sphere at the bottom, where the 2 men would sit, was made of steel 5 inches thick. The window was a cone of plexiglass so narrow that you had to press your face against it just to look out. They began their descent at 8 in the morning. It took nearly 5 hours to reach the bottom. 5 hours of slow, cold, total darkness. The temperature outside dropped toward freezing. Occasionally the steel groaned as the pressure increased. At one point, a sharp crack rang through the sphere, loud enough to make both men think the vessel was failing. They checked their instruments. Everything held. Later they discovered it was one of the plexiglass windows in the entry tunnel cracking under the pressure. They kept going. At 1:14 in the afternoon, the Trieste touched bottom, 7 miles down, 35,800 feet, the deepest point any human being had ever reached. And Picard pressed his face to the tiny window. On the sediment outside, stirred up by their landing, a flatfish drifted in the floodlights. A fish, at the bottom of the Mariana Trench, alive. Picard later wrote that in that moment he had the answer biologists had been asking about for decades. Could life exist at the greatest depths of the ocean? The flatfish said yes. Clearly, obviously, unmistakably yes. They had 20 minutes at the bottom, 20 minutes after 5 hours of descent. They could not take photographs. The silt their landing had stirred up clouded the water around them. They recorded what they could, dropped their iron ballast to begin rising, and spent another 3.5 hours climbing back to the surface. The world now knew the bottom of the ocean was not dead. But knowing something lives there and knowing what lives there, how it survives, and why it is built the way it is, those are entirely different questions. And answering them required technology that did not exist yet. The Trieste cracked one window just getting down there. Something far more sophisticated was needed. It would take science another half-century to build it. 500 feet below the ocean surface, the last photon of sunlight disappears. 500 feet. That is roughly the height of a 40-story building. In the scope of an ocean that reaches 36,000 feet in places, sunlight barely grazes the top. Everything below 500 feet lives in permanent darkness. Not the kind of dark you get in a room with the lights off, where your eyes adjust and shapes slowly appear. The deep ocean dark is absolute. The biological dark. The kind where your eyes receive zero input at all, because there is zero light to receive. This creates an immediate problem for any creature that evolved from surface life and then moved deeper. Vision, as most animals use it, requires light bouncing off objects and into an eye. Remove the light, and vision becomes useless. Eyes that serve no purpose cost energy to maintain. So over millions of years of evolution, creatures that moved into the deep ocean either lost their eyes entirely, developed eyes so large they could collect the faintest possible trace of light, or found another strategy entirely. Some deep-sea fish have eyes the size of dinner plates relative to their body. They are tuned to catch the last scraps of light filtering down from above, mostly blue light, which penetrates deeper than any other wavelength. Other animals developed organs that sense movement or vibration instead of light. The lateral line system, a row of pressure-sensitive cells running along the body, lets a fish feel the water displacement of another creature moving nearby, even in total darkness. But the most spectacular solution is one where the animal stops needing external light at all. It makes its own. This is bioluminescence, and in the deep ocean, it is everywhere. Scientists estimate that over 90% of deep-sea creatures produce their own light through chemical reactions inside their cells. Jellyfish glow blue. Anglerfish dangle a light in front of their face like a lantern on a hook. Some squid flash patterns at each other in what appears to be a form of communication. Down in the Hadal zone, past 15,000 feet, even bioluminescence becomes rare. The creatures there live in a world where no light has existed for millions of years. Their biology has adapted so completely to the absence of light that the snailfish, the dominant predator of the Mariana Trench, has no functional eye pigment. It has lost the genetic machinery for seeing colour entirely, because colour requires light, and light does not exist where it lives. But losing something is only half the story. The other half is what these animals gain to replace it, because something stranger than vision is happening down there in the permanent dark, and it only becomes clear when you look at how the animals eat. In the deep ocean, the most dangerous thing you can do is be visible. There is no sunlight to hide in, no shadows to blend into. The darkness is total, and the only light that exists is the light a creature makes itself, which means that in the deep sea, being bioluminescent is both a weapon and a target. Imagine you're a small fish, 6,000 feet down. You have no eyes that work. You sense the world through vibration and chemical signals in the water around you. Then, ahead of you, a faint blue glow appears. It pulses rhythmically. It looks, to the limited sensory apparatus you have, like something small and edible drifting in the current. You move toward it. It is a trap. The glow is attached to a long, flexible spine growing out of the forehead of an anglerfish 3 times your size. The moment you get close enough, the anglerfish opens its mouth, and the current it creates sucks you straight in. This is one of the most elegant predator designs in the history of life on Earth. The anglerfish grows its own fishing rod, complete with a glowing lure, and uses it to bring prey directly to its mouth in the dark. The bioluminescent tip is not a random feature. It is a precisely tuned biological tool, bright enough to attract prey, dim enough not to attract larger predators from too far away. But bioluminescence is not only used for hunting. Some creatures use it as a distress signal, flashing a burst of light when attacked to startle a predator and buy time to escape. Others use it as a form of counter-illumination. When viewed from below, a creature silhouetted against the faint glow of water above it is visible. Some deep-sea fish produce light from their belly in exactly the pattern of the ambient glow, making them effectively invisible from below. Scientists have identified dozens of distinct uses for bioluminescence across deep-sea species, and they believe this is only the fraction they have managed to observe directly. The deep ocean is so vast and so hard to reach that most of the creatures down there have never been seen by a human being. What is strange about bioluminescence chemically is that it evolved independently in dozens of different animal lineages. Fish, squid, jellyfish, worms, and bacteria all arrived at the same solution through entirely different paths. This is called convergent evolution. The deep ocean is so punishing and the need for light so pressing that natural selection kept arriving at bioluminescence no matter what species it was working with. In the Hadal zone, below the range where even bioluminescence is common, something even more unusual takes over. The creatures there do not rely on light at all. They sense the world through other means entirely, and their bodies reflect this. They are pale, often translucent. Their tissue almost colourless because colour, like vision, became useless. Some of these animals have not been studied at length because getting them to the surface alive is nearly impossible. The pressure change kills them on the way up. Scientists sometimes only get to examine a creature after it is already dead. That problem changes everything about what we know and what we are still guessing at. Of all the creatures living in the Mariana Trench, one is stranger and more compelling than anything you would expect to find at the bottom of the ocean. Its name is the Hadal snailfish. Its scientific name is Pseudoliparus swirei, and scientists currently hold the record for the deepest fish ever observed. Cameras have captured it at roughly 5 miles down, deeper than any other vertebrate on Earth has ever been confirmed alive. It does not look like a predator. It looks like something that got lost. The Hadal snailfish is soft, pale, and translucent. Its skin is so thin you can see its internal organs from the outside. It has no scales. Its body is mostly water and fat. It moves slowly, drifting through the sediment in tight groups, hoovering up small crustaceans with a mouth that looks almost gentle. It is the top predator of the Hadal zone, the apex of a food chain that operates in total darkness, under pressure that would kill everything living on land, feeding on creatures that themselves survive by eating chemicals seeping from rock. What makes this fish scientifically extraordinary is the genome. Scientists sequenced its complete set of genes and found a series of changes unlike anything seen in surface fish. First, the gene that normally controls bone hardening has a specific mutation that prevents the skeleton from fully mineralizing. The Hadal snailfish has bones that are partly soft cartilage, like a baby's. This is not a defect, it is a design feature. Rigid bone under extreme pressure would crack. Soft cartilage bends. Second, its cell membranes are unusually rich in a type of fat that stays fluid at high pressure. Most fats would solidify under these conditions, and a cell with a solidified membrane cannot function. The snailfish evolved a specific fat composition that keeps its cells flexible even when squeezed by the weight of 6 miles of ocean. Third, its skull is not fully closed. The bones of its head are connected loosely with gaps. In a surface fish, this would be a serious structural weakness. Down here, with no predators larger than itself, there is no evolutionary pressure to have a hard skull. This soft, open architecture is cheaper to maintain and more pressure tolerant. The Hadal snailfish split from its nearest shallow water relative roughly 20 million years ago. Since then, it has been evolving in isolation in the dark, under crushing pressure, with almost no food available. The result is an animal that looks fragile but is built for one of the most hostile environments on the planet. When scientists tried to bring specimens to the surface for study, the fish died during the ascent. The pressure change that should have relieved stress on the animal instead killed it. It adapted so completely to high pressure that normal surface conditions were lethal. The snailfish is not adapted for survival everywhere. It is adapted for survival in one precise place, the bottom of the world. Change the environment by even a small amount, and it dies. That specialization is what makes the next discovery so astonishing. Because the snailfish is not the strangest thing living down there. Every protein in your body has a specific three-dimensional shape. That shape is what makes it work. An enzyme that digests food is shaped like a lock. The molecule it processes fits into that lock like a key. Change the shape, and the lock does not work. The enzyme fails. If enough enzymes fail, the cell dies. If enough cells die, the organism dies. At high pressure, protein shapes distort. The forces acting on the molecule from outside squeeze and warp the structure. This is one of the central challenges of life in the deep ocean. Everything runs on proteins. Pressure breaks proteins. How does anything survive? In the 1980s, a marine biologist named Paul Yancey started noticing something unusual in deep-sea fish. Their muscles contained high concentrations of a compound called trimethylamine N-oxide, a small organic molecule that scientists had mostly considered irrelevant before. In deep-sea fish, it was present in amounts far higher than in surface fish. Yancey ran experiments. He found that this compound, added to a solution containing proteins, protected those proteins from deforming under pressure. It acted like a molecular scaffold. The water molecules around a protein normally form a structured network. Under high pressure, that network distorts, pulling the protein out of shape with it. This compound kept the water network stable. It kept the proteins folded correctly. It was a chemical solution to a physics problem. Life had invented a molecule that fought back against the crushing force of the deep ocean, one protein at a time. The concentration of this compound in fish tissues rose predictably as you went deeper. Shallow fish had almost none. Fish at 3 miles down had moderate amounts. Fish at 6 miles down were saturated with it. The pattern was so consistent it became a way of predicting how deep a fish lived just by analysing its muscle tissue. Then in 2025, a team doing the most comprehensive biological survey of the Hadal zone ever attempted found something that broke that pattern. Below 6 miles, fish did not have higher concentrations of this compound. They had the same amounts as fish at shallower depths. The expected increase stopped. Scientists are still working out why. One possibility is that the compound reaches a concentration limit, beyond which adding more does not help and may even be harmful. Another is that organisms at the very deepest levels have evolved a completely different mechanism for coping with pressure, one that does not depend on this molecule at all. What this means is that our model of how deep sea life survives is incomplete. The rules we thought applied across the entire Hadal zone may only apply to part of it. Something else is happening at the very bottom, something science has not fully identified yet. And the deeper you go into what else lives down there, the more this sense of incomplete knowledge starts to feel like a weight of its own. The following is about a single cell. One cell. No brain. No nervous system. No heart. No organs of any kind. This single cell is about 4 inches wide. A typical cell in your body is so small it is invisible without a microscope. Thousands of them could fit on the head of a pin. The cells that make up your skin, your liver, your heart, all of them are microscopic. So a cell the size of a piece of fruit seems scientifically impossible. And yet, on the floor of the Mariana Trench, they sit in the sediment like boulders. Scientists discovered them using cameras dropped to the seafloor and were genuinely confused at first about what they were looking at. These organisms are called xenophyophores. They are the largest individual cells of any organism on Earth, and they are doing something remarkable with that size. A xenophyophore looks something like a pale, irregular blob anchored to the sediment. It has no defined shape. It extends branches through the surrounding mud, gathering particles of organic material, dead bacteria, tiny specks of food drifting down from above. It processes what it finds and incorporates the waste products back into its own body, building up a kind of skeleton made from fecal pellets and inorganic particles cemented together with its own biological glue. This organism lives its entire life as a single cell, grows to the size of a mango, builds itself a structure out of its own waste, and contributes directly to the recycling of nutrients on the ocean floor. Scientists believe xenophyophores play a significant role in the chemistry of the deep-sea sediment. The question that naturally follows is why? Why be so large? One answer is surface area. A larger cell can collect more food from the surrounding water. In an environment where food is extremely scarce, bigger may be better. Another answer is pressure. Large, soft cells may actually handle pressure better than small, rigid ones under certain conditions. The physics of compression works differently at different scales. But here is the thing, xenophyophores have been found deeper in the ocean than almost any other large organism. They do not just survive at extreme depth, they appear to prefer it. The deeper you go, the more of them you find. A single cell that thrives where almost everything else struggles. A single cell that reaches the size of your fist. A single cell that may be fundamental to how the deep ocean floor processes the nutrients that eventually cycle back to the surface. One cell, doing all of that. And it barely gets mentioned when people talk about the Mariana Trench, because the creatures down there keep getting stranger the more you look. Every ecosystem on Earth's surface runs on the same fuel, sunlight. Plants capture it. Animals eat the plants. Other animals eat those animals. The entire chain, every cow, every insect, every person, traces its energy back to a star 93 million miles away. Then scientists discovered something that broke this rule entirely. In the deep ocean, in places where no sunlight has ever reached, entire communities of living things are powered by a completely different process. They do not need the sun. They eat chemicals. The process is called chemosynthesis. It works like this. Certain bacteria can take molecules like hydrogen sulfide or methane, chemicals that are toxic to most life, and use them as fuel instead of sunlight. These bacteria pull chemical energy out of the reaction and use it to build organic compounds. They become the base of a food chain just like plants are at the surface, except instead of sunlight driving the process, it is the chemical energy locked inside poison gas seeping from the Earth's crust. This discovery happened first at hydrothermal vents in the late 1970s, when scientists found dense communities of tube worms and clams thriving near superheated water venting out of the ocean floor. The vent water was loaded with hydrogen sulfide. The animals around it were using chemosynthetic bacteria, either living in their own tissues or forming the base of the food web, to convert that poison into energy. The Mariana Trench has cold seeps rather than hot vents. In these seeps, methane and hydrogen sulfide leak slowly and steadily through cracks in the rock. The temperature is near freezing. There is no dramatic plume of superheated water, just a slow, cold seeping of chemicals from below. And around those seeps, life clusters. Tube worms anchor themselves to the rock. Clams settle into the sediment. Bacteria form thick mats over the seafloor. The chemical energy coming up from the crust below is the fuel. The bacteria are the engine. The worms, clams, and everything above them in the food chain are the result. This is not a minor discovery. This is a complete alternative model for how life is powered. For billions of years, the assumption was that life needs a star. Now we know that life needs energy, and energy can come from inside a planet just as well as from the sky above it. What this means for life elsewhere in the universe is something we will get to. But first, we need to look at what those seep communities actually look like up close. Because when a Chinese submersible finally documented them in full detail, the discovery was bigger than anyone expected. In July 2025, a scientific paper published in the journal Nature described something that had never been documented before. A team of scientists operating a manned submersible called Fenduji dove into two ocean trenches in the northwest Pacific, the Kuril-Kamchatka Trench and the Aleutian Trench. What they found stretched across 1,500 miles of ocean floor at depths between about 3.5 and almost 6 miles down. A garden of sorts. Dense communities of tube worms and clams carpeted the seafloor near cold seeps. The tube worms, a species related to the tube worms found at shallow hydrothermal vents, were anchored to the rock in thick clusters. The clams packed the sediment in dense beds. Bacteria formed mats over every available surface. This was not a small pocket of life. The chemosynthetic communities they documented were, according to the paper, the deepest and most extensive ever found on Earth. Let that scale land for a moment. The worm communities alone covered spans of seafloor larger than many city blocks, at depths where the pressure is so extreme that a human would be killed before understanding what was happening. The tube worms are extraordinary creatures on their own. They have no mouth, no digestive system at all in the traditional sense. Instead, their bodies contain a specialized organ packed with chemosynthetic bacteria. The worm absorbs hydrogen sulfide and oxygen from the water and passes them to the bacteria. The bacteria produce organic molecules. The worm lives off those molecules. The entire digestive function is replaced by a partnership with a microorganism. The clams in these communities also host chemosynthetic bacteria in their gills. Both the worms and the clams have over evolutionary time essentially given up processing their own food and outsourced digestion to bacteria. This kind of biological outsourcing, where one organism does a critical job for another, is called symbiosis. At the deep-sea seeps, it is the fundamental building block of everything, and the communities found in 2025 were the deepest ever recorded. But the scientists noted something even more important in their paper: the fact that chemosynthetic communities this large existed in two separate trenches suggested they were probably common across many trenches worldwide. They had simply never been looked for carefully enough. The deep ocean is not a barren desert between a few hotspot vents. It is a landscape dotted with chemical gardens, spread across the floor of every ocean on Earth, sustained by energy coming from inside the planet itself. Nobody knew this a decade ago. Most of the Hadal Zone has no cold seeps, no tube worms, no chemical gardens, just sediment and falling particles. The floor of the deep ocean is covered in a layer of fine material that accumulates so slowly you could watch it for a year and see almost no change. Scientists call this accumulation marine snow, and it is the primary food source for most of the life in the Hadal Zone. Marine snow is not actually snow. It is a constant, slow drift of dead and decaying organic material falling from the sunlit surface layers above. Dead bacteria, microscopic algae, the fecal pellets of small crustaceans, fragments of dead fish and whale falls. All of this sinks slowly through thousands of feet of dark water. The journey from surface to Hadal zone takes weeks to months. Most of the organic material gets consumed on the way down by bacteria in the mid-water zone. By the time anything reaches 6 miles down, it has been picked over by microbes for months. What arrives at the bottom is a thin, poor-quality food source. Not much nutrition, not much of it. This is why most animals in the Hadal zone are small, slow, and energy efficient. There is no evolutionary advantage to being fast or large when your entire food budget for a year might arrive in a thin sprinkle of dead particles. The snailfish moves slowly. The amphipods are small. The microbes in the sediment are adapted to eat things that surface microbes would not bother with. But marine snow is not consistent. Occasionally something much larger arrives. A dead whale sinks to the floor and becomes an event of enormous biological importance. The sudden arrival of a whale carcass, thousands of pounds of protein and fat, is like a meteor of nutrition in a food desert. Every scavenger in the vicinity converges. Sharks, fish, worms, bacteria, all of them feeding in layers, processing the carcass through a succession of different communities that can last for decades. Scientists have found whale falls on the deep ocean floor and identified distinct phases of decomposition, each hosting a different community of specialized organisms. Some of the species found on deep-sea whale falls have been found nowhere else on Earth. There is also something deeply strange about how marine snow accumulates in trenches specifically. Trenches act as collection points. Their slope-walled structure funnels sediment inward and downward. The very bottom of a trench is richer in organic material than the surrounding flat seafloor, simply because of gravity. The Hadal zone is a trap for everything that falls, which means the deepest point of the ocean is, counterintuitively, one of the richer spots in the abyss. And the creatures that exploit this concentration most effectively are not the fish, or the worms, or the microbes. They are a species of tiny, almost invisible crustacean that can arrive in overwhelming numbers and strip a food source down to nothing in hours. Drop a piece of dead fish into the Mariana Trench and wait. Within hours, something will find it. Amphipods are shrimp-like crustaceans that measure between half an inch and a couple of inches long. They look like curled-up sow-bugs, pale and nearly see-through. Individually, each one is unremarkable. Together, they are one of the most efficient scavenging systems in nature. Scientists have used baited traps to study deep-sea scavengers, and the footage from these traps is striking. A piece of bait placed on the ocean floor at Hadal depths attracts amphipods within hours. Within a day, the bait is buried under a writhing mass of them, sometimes thousands of individuals, all feeding simultaneously. In footage from traps in the Mariana Trench, the water around the bait becomes almost opaque from the density of amphipods. They are the cleanup crew of the Hadal zone. In an environment where food is extremely scarce, waste is not an option. When something dies, or when a large piece of marine snow reaches the floor, amphipods are among the first to arrive and among the most thorough at processing it. Their bodies are built for this. Amphipods have compound eyes that detect minute traces of chemical signals in the water, the scent of decomposing organic material. They can detect these signals from long distances relative to their size, allowing them to navigate across the featureless dark sea floor toward a food source they cannot see. Some amphipod species in the Hadal zone have been found at extraordinary densities. In one study, a trap pulled up from the deepest part of the Mariana Trench contained over 300 individuals from a single overnight deployment. These are not rare creatures struggling to survive. They are abundant, well adapted, and thriving. Amphipods also occupy a central position in the Hadal food web. They eat the marine snow and the decomposing matter on the seafloor, converting it into amphipod tissue. Then the snailfish eats the amphipods. The snailfish is the top predator. It eats almost nothing else. This creates a surprisingly tight chain. Marine snow feeds amphipods. Amphipods feed snailfish. Snailfish live at the deepest fish depth possible, Remove any link and the whole Hadal ecosystem shifts. But the amphipods are also the link that connects one of the most disturbing discoveries ever made about the deep ocean. When scientists examined the stomach contents of amphipods from the Mariana Trench, every single one contained something that had no right to be there. Something that was not marine snow. Something that was not dead bacteria or bits of fish. Something human. In March 2012, James Cameron climbed into a submersible he had helped design, sealed the hatch, and began sinking toward the bottom of the Mariana Trench alone. Cameron is best known for directing Titanic and Avatar. But his obsession with the deep ocean predated both films. He had been thinking about a solo dive to Challenger Deep for years, and the vessel he co-designed, the Deep Sea Challenger, was the result of that obsession made physical. The Deep Sea Challenger was shaped like a vertical torpedo rather than the flattened submarines most people imagine. This shape allowed it to descend faster, reaching the bottom in about 2.5 hours rather than the 5 hours it took the Trieste in 1960. Cameron sat in a sphere of high-strength steel 7 feet in diameter at the bottom of the craft, surrounded by monitors and scientific equipment. He touched down on the floor of Challenger Deep alone, 6.8 miles below the surface of the Pacific Ocean, the first person to make that journey solo. He spent about 3 hours on the bottom. He collected sediment samples. He deployed scientific instruments. His cameras recorded footage of the seafloor in high definition, the first such footage ever captured from the deepest point on Earth. And what he saw was, by his own account, deeply strange in one particular way. It was almost empty. The seafloor he landed on was flat, featureless, and grey. Very little moved. Some pale shapes in the sediment, possibly sea cucumbers, shifted slowly. Almost no fish. Almost nothing visible on the surface of the sediment at all. This confused scientists initially. The expectation, based on what was known about other deep-sea environments, was that the floor of the trench would at least have some visible macrofauna. But Challenger Deep, the deepest of the deep, appeared nearly lifeless on first visual inspection. The apparent emptiness had an explanation, but it took subsequent missions to find it. Life at the very bottom of Challenger Deep is mostly microbial. The bacteria and archaea in the sediment are there in extraordinary density, but they are invisible to cameras. The macroscopic creatures are rare, scattered, and often stay buried. Cameron's dive proved the ocean floor could be reached regularly and safely with modern technology. It helped open the door to far more systematic scientific exploration. But the mission that would change everything happened a decade later, using a different approach entirely. Less camera time, more biology, more sampling, and one discovery that nobody had put on their list of expected findings. In 2021, a team of Chinese scientists boarded the manned submersible Fenduje at a dock in China and began a series of dives into the Mariana Trench that would continue over the next several years. Over dozens of individual dives, the team collected thousands of samples: sediment from the trench floor, seawater from different depths, biological material scraped from rock. They brought these samples back to laboratories and sequenced the genetic material inside them. The results, published in the scientific journal Cell in March 2025, describe something the scientific world had not fully seen before. Over 7,000 microbial species identified from the Hadal zone, the vast majority of them previously unknown to science, each one genetically distinct, each one adapted in its own way to the specific conditions of the deep. 7,000 species of microorganism living in one of the most extreme environments on Earth, most of them never described, never named, never even suspected. The microbial diversity in the Mariana Trench is, on a genetic level, extraordinary. Scientists found species of bacteria and archaea, a second type of single-celled organism distinct from bacteria, adapted to every specific niche of the trench environment. Some lived in the sediment, some lived in the water column, some were found attached to the surfaces of particles falling from above, some appeared to live primarily in the bodies of other organisms, Many of these microbes had genes that surface organisms simply do not have. Genes for processing specific chemicals, genes for repairing damaged proteins under high pressure, genes for extracting energy from molecules that would be nutritionally useless to any surface-dwelling organism. The genetic diversity found was compared to the diversity you might expect across completely different ecosystems on the surface, not within a single trench. The Hadal zone had diversified, over millions of years of isolation, into a biological world as complex as a forest. And buried within this diversity was a practical discovery that researchers flagged immediately. Some of the microbial genes found had structures and functions with direct potential applications in medicine and biotechnology. Deep-sea microbes that survive extreme pressure have evolved proteins that are unusually stable, unusually resistant to breakdown, unusually efficient at their functions. These properties are exactly what chemical engineers and drug designers look for when developing industrial enzymes and medical treatments. The bottom of the ocean might also be a library of biological tools that human science has not yet learned to read. Most of the genetic changes that make deep-sea microbes unusual come back to one central problem: high pressure destroys molecular structure. At 6 miles down, the pressure acting on every cell in every organism is enormous. Every protein that folds, every membrane that flexes, every enzyme that processes a chemical reaction, all of them must work correctly under conditions that would prevent them from functioning at the surface. The microbes in the Mariana Trench have been evolving solutions to this problem for millions of years, and their genomes show the record of that evolution in remarkable detail. Some species found in the deepest sediment have what scientists describe as streamlined genomes. They have fewer genes than their surface relatives. Every gene that serves a function that is irrelevant at depth has been cut away. The result is a genome that is lean, efficient, and specialized for one exact environment. Other species went the opposite direction. They have larger, more flexible genomes with extra copies of certain key genes, particularly genes involved in repairing damaged proteins and rebuilding membranes. Under constant pressure stress, DNA itself can be damaged. These microbes carry duplicated repair systems, redundancy built into their genetics, so that if one system fails, another takes over. Scientists found one specific pattern across multiple unrelated species. Genes for handling pressure tended to cluster together on the genome, as if they evolved as a unit. Species that lived deeper had more of these clusters. The correlation between depth and the presence of these genetic packages was strong enough to be statistically striking. What this says about evolution is significant. Given the same extreme environmental pressure, applied over millions of years, completely unrelated organisms converge on the same genetic solutions. The deep ocean is testing life with the same exam, and life keeps writing the same answers. Among the genes found was a set specific to processing polyunsaturated fats, the same type of fat that keeps cell membranes fluid under high pressure. Multiple unrelated microbial species have developed expanded gene families for making these fats, entirely independently of each other. The 2025 study also found something unexpected in the carbon cycle. Methane-consuming bacteria in the trench sediment are actively processing and converting methane that seeps up from the Earth's crust. Methane is a powerful greenhouse gas. These microbes are consuming it at depth before it ever reaches the atmosphere. The scale of this process at a global level is still being calculated, but the possibility that deep-sea microbes are playing an active role in preventing methane from entering the atmosphere, a role nobody knew about before the Mariana Trench research, is one of the most significant environmental discoveries buried in these findings. What happens underground and under the sea matters for the air above it, and what these microbes were eating and what was already inside the animals they lived alongside was about to reveal a chapter of this story that has nothing to do with biology. It has to do with us. In 2018, a team of scientists sent baited traps down into 6 of the deepest ocean trenches in the Pacific. Their goal was simple: catch amphipods, bring them up, and study their biology. When the traps came back up, The scientists opened the amphipods and looked inside their digestive systems under a microscope. Every single amphipod from the Mariana Trench had plastic fibres in its gut. Every one. 100% of the specimens collected from the deepest place on Earth had already eaten human-made plastic before a scientist had ever laid eyes on them. The plastic was not in big pieces. It was microplastic. Fragments and fibres smaller than a grain of rice, broken down from larger objects by years of exposure to salt water and sunlight before sinking. Once small enough, these particles drift downward through the water column just like marine snow. They reach the bottom, they get mixed into the sediment, the amphipods eat the sediment, the plastic goes with it. The trench acts as a funnel, its steep walls direct falling material inward and downward, concentrating everything that sinks at the very lowest point. This means the Mariana Trench, the deepest and most remote place on the surface of the Earth, is also one of the most efficient collection points for everything humanity drops into the ocean. A plastic bottle thrown into the ocean off a beach in California does not stay near the surface forever. It breaks apart. It sinks. Years later, a fragment of it may sit in the gut of a creature 6 miles below the surface, at a place no human has ever visited, eating something no human designed as food. The types of plastic found in the amphipods were recognizable: nylon, polyethylene, polyester. The same materials used in clothing, packaging and water bottles. The same materials filling landfills and washing into rivers and eventually into the sea. The study looked at amphipods from 6 different trenches across the Pacific and found plastic in over 70% of all individuals examined. In the Mariana Trench specifically, the rate was 100%. What this does to amphipods biologically is still being studied. Plastic particles can carry chemical contaminants. They can block digestive passages. They can create a false sense of fullness, reducing how much actual food an animal consumes. In surface ocean animals, the effects have been documented in detail. In the Hadal zone, scientists are only beginning to understand the consequences. But the biological question is almost secondary to the geographical one. The fact that plastic is in every amphipod at the bottom of the Mariana Trench means that human pollution has reached every corner of this planet. Not most corners, every corner. 6 miles down in crushing darkness, in an environment so hostile that building a vessel to reach it cost tens of millions of dollars. Human waste was there first. The scale of what that represents is hard to sit with, and one discovery made it impossible to look away. In 2020, scientists announced the discovery of a new amphipod species found in the Mariana Trench at nearly 4.5 miles down. When a new species is discovered, the scientists who find it get to name it. The name they chose for this one was deliberate and uncomfortable and worth understanding. They named it Eurythanes plasticus. The second word is the genus name for a group of deep-sea amphipods. The third word refers to plastic. They named a newly discovered species after the plastic found inside its body. The plastic found in this amphipod was a compound called polyethylene terephthalate. You know it by a different name. It is the material used to make water bottles and synthetic workout clothing. The specific plastic inside this creature, living nearly 4.5 miles beneath the Pacific Ocean, was a material manufactured by humans for consumer products. The scientists who named the species were explicit about why they made that choice. They wanted to make it impossible to discuss this animal without discussing what was inside it. They wanted the contamination to be built into the scientific record permanently. This means that in every future scientific paper that mentions this species, in every database entry, in every museum catalogue, the word plastic will appear. Long after the people who named it are gone, the name will carry the record of what was found inside the first specimen. Heike Vesper of the World Wildlife Fund noted at the time that this species had plastic in its body before humanity even knew it existed, before it had a name, before it appeared in any scientific record, before any human had ever seen it alive. It had already eaten something we threw away. That timeline matters. The plastic entered the trench before the science did. The contamination predates the discovery. This raises a question with no clean answer. How many species in the Hadal zone have already been altered by human pollution before we have documented them? How many have shifted their diet, their body chemistry, or their behavior in response to material we introduced to their environment without knowing they existed? The deep ocean was already the least explored place on Earth. Now it may also be a record of ecological change that happened in the dark, unseen, before anyone thought to look. And the person who went down to look, and who saw what else was there, found something that made the scientific case land differently, because he found the plastic himself, with his own eyes, in person. On April 28, 2019, a retired United States Navy officer named Victor Vescovo became the deepest human being who has ever lived. He descended to 35,853 feet in a submersible called the DSV limiting factor, nearly 60 feet deeper than James Cameron's 2012 dive. The measurement was independently verified. It stands as the confirmed deepest point any human being has ever reached. Vescovo spent 4 hours on the floor of Challenger Deep. He moved across the sediment. He observed creatures. Scientists analysing the data from his dive later identified what appeared to be a new species of crustacean. He also saw a plastic bag and candy wrappers at 35,000 feet, at the deepest point of the deepest trench of the deepest ocean, at a place that requires tens of millions of dollars of engineering and years of preparation to reach. A plastic bag and candy wrappers were already there. Vescovo was not the first to see plastic at depth, but his dive made the reality of it impossible to distance from. He was a human being, sitting in a sphere of steel, looking through a window at the most remote floor on Earth, and the view included human garbage. The DSV limiting factor was a remarkable machine. Unlike the Trieste, which took 5 hours to descend and could only carry 2 people, or the Deep Sea Challenger, which was a one-person craft with limited scientific capability, the limiting factor was designed for repeated use. It made multiple dives to Challenger Deep in 2019 as part of Vescovo's 5 Deeps expedition, reaching the deepest point in each of the world's 5 oceans. The scientific payoff from these repeated dives was significant. Sediment samples, water samples, biological specimens, high-definition footage, temperature and pressure measurements. The data collected was shared with research institutions worldwide, but no piece of data from those dives carried the same immediate weight as the photograph of a plastic bag sitting on the floor of Challenger Deep. We mapped the moon before we mapped the ocean floor. We sent rovers to Mars decades before sending a camera to the deepest point of our own planet. And when we finally got there, in person, with our own eyes, we found evidence that we had arrived before, in the worst possible way. Something found in the acoustic record of the trench was also waiting for an explanation, and for over 2 decades, nobody could agree on what it was. On May 1st, 1997, a hydrophone array operated by the National Oceanic and Atmospheric Administration picked up an ultra-low frequency sound in the Pacific Ocean. A hydrophone is an underwater microphone designed to detect sounds that travel through seawater. The National Oceanic and Atmospheric Administration operates a network of them across the Pacific, originally built during the Cold War to track submarines. By the 1990s, scientists were using this network to listen for natural sounds in the deep ocean. The sound recorded that day was unusual. It rose rapidly in frequency over about 1 minute, then cut off. It was extraordinarily loud enough to be detected by hydrophones over 3,000 miles apart. No known geological or biological source matched its profile. Scientists nicknamed it the bloop. The bloop became one of the most famous unexplained sounds in oceanography. For years, speculation ran wide. Some suggested it could be a large unknown biological organism. Some pointed out that the frequency profile and power of the sound was, in raw terms, consistent with what a very large animal might produce. The ocean is mostly unexplored. Something enormous could theoretically exist in the deep that science has not yet found. This idea spread well beyond scientific circles. The bloop became a touchstone for people interested in the possibility of unknown creatures in the deep. Then, in 2005, scientists took a closer look at the full acoustic profile of the sound and compared it against a better understood phenomenon: the fracturing and calving of large ice masses in the Antarctic and Arctic. Large blocks of ice cracking apart produce extremely loud, low-frequency sounds. The acoustic signature of the bloop, when analysed against higher resolution data, matched ice fracture patterns closely. The National Oceanic and Atmospheric Administration updated their assessment. The bloop was almost certainly a massive Antarctic ice event, a sound produced by a glacier or ice shelf breaking apart, transmitted thousands of miles through the deep sound channel of the Pacific Ocean. The deep sound channel is a layer of water at a specific depth, where temperature and pressure combine to create conditions that allow sound to travel extraordinary distances with almost no loss of energy. Whales use it to communicate across entire ocean basins. Human-made sounds travelling through it can be detected on the other side of the planet. The deep ocean is not silent. It is one of the most acoustically complex environments on Earth. Earthquakes, whales, ship propellers, ice. All of it travelling through water with a clarity and range that no surface environment matches. And what the acoustic record of the Mariana Trench itself sounds like is something almost nobody talks about. In 2021, the Chinese research vessel Tansuozhe set course for the Mariana Trench with a crew of scientists and a manned submersible aboard called Fenduje, which translates loosely to striver. Over the following years, the Fenduje completed 33 dives into the Hadal Zone, reaching depths approaching 6 miles. The scientists aboard collected sediment cores, water samples, rock samples, and biological material. They deployed instruments that stayed on the seafloor for months at a time, transmitting data back through acoustic modems. This was the most comprehensive biological survey of the Hadal Zone ever conducted. Previous deep-sea expeditions had been snapshots, a few hours on the bottom, a handful of samples. The Fenduje mission was a systematic campaign to document an entire ecosystem from top to bottom, over time, across multiple sites. The scope of what they brought back transformed the scientific understanding of the Hadal Zone in a way that no single dive had managed before. The 7,000 microbial species, the chemosynthetic communities, the unexpected patterns in pressure adaptation molecules, the methane-consuming bacteria, the full food web analysis, all of it came from this mission. The study published in Cell in March 2025 was the flagship paper from this work, but it represented only a portion of the total scientific output. Dozens of additional papers are still being written from the samples collected. One of the most operationally significant findings was how much life existed in the water column above the trench floor, not just at the bottom. Microbial communities at different depths showed distinct compositions, with different species dominating at different pressure levels. The trench is not just a floor with creatures on it. It is a layered environment with different ecosystems stacked on top of each other from the rim to the bottom. This means that the question of what lives in the Hadal Zone is not a single question. It is dozens of questions, one for each depth layer, each with its own set of species, adaptations and ecological dynamics. The Fenduje mission began to answer some of them, but every answer found more questions underneath. And one of those questions, sitting in the data about methane-consuming bacteria, pointed in a direction nobody had been looking. When the Nature paper published in July 2025 landed in the scientific community, the reaction was immediate. Scientists using the Fenduje had discovered chemosynthetic communities, the cold seep ecosystems of tube worms and clams running on chemical energy, at depths nobody had confirmed before, nearly 6 miles down in some cases, stretching across more than 1,500 miles of ocean floor between 2 separate trenches in the Northwest Pacific. To understand why this mattered so much, you need to know what scientists expected to find before they looked. The prevailing picture of the Hadal zone was one of extreme scarcity. The cold seep communities found at shallower depths were known to exist at around 3 to 4 miles down. Below that, the assumption was that conditions became too extreme even for chemosynthetic life. The pressure too high, the temperature too cold, the chemical gradients too weak. The Fendougé mission showed that picture was wrong, not slightly wrong. The deepest confirmed chemosynthetic communities found on this expedition were nearly 1.5 miles deeper than previous records. The assumption about a lower limit for complex life in the Hadal zone was not a rough approximation. It was a significant underestimate. The tube worms observed at these depths were recognizable relatives of species found at shallower seeps, but genetically distinct, adapted over evolutionary time to conditions significantly more extreme than their cousins face. The clams were similarly diverged from their shallower relatives. What the discovery says scientifically is that chemosynthetic life is more adaptable than anyone thought. It can push deeper. It can colonize environments at the edge of what biology can technically achieve. And because it runs entirely on chemical energy from the Earth's interior, it is not limited by the distance from the surface or the availability of sunlight. This is the key insight that keeps repeating throughout the story of the Mariana Trench. Life does not need what we assumed it needed. The rules that scientists wrote for the requirements of life, energy from the sun, moderate temperatures, accessible chemistry, were written by studying life in the environments they had access to. Environments near the surface, environments with light. The deep ocean has been running a completely different experiment for hundreds of millions of years, and the results of that experiment are still coming in. One of the most striking results involves not what lives in the trench, but what the floor of the trench actually looks like, because most people imagine it as still, frozen, a motionless plain of sediment. They are wrong. Remove sunlight from an ecosystem and it does not collapse. It reorganizes. The Hadal food web is one of the most stripped down biological systems on Earth. Every link in it is visible if you look closely, and every link tells you something about the physics of extreme environments. At the base, 2 parallel systems provide energy. Marine snow, the slow fall of organic particles from above, delivers carbon that has already passed through dozens of organisms on the way down. By the time it reaches the trench floor, it is thin, degraded, and poor in nutrition. Chemosynthesis, at the seeps, provides a richer, more concentrated energy source for the communities lucky enough to be near a seep. Microbes process both of these inputs at the base level. Bacteria and archaea in the sediment consume organic particles and chemical compounds, breaking them down and releasing their energy. The microbial layer is invisible from the surface, but constitutes the majority of biological activity in the trench by mass. Above the microbes, small invertebrates feed on the sediment and the microbial community within it. Polychaete worms, bristleworms, move through the sediment ingesting material. Sea cucumbers drag themselves across the floor, processing sediment through their digestive systems and extracting nutrients. Isopods and amphipods scavenge any concentrated food source they can find. The amphipods occupy a crucial middle position. They are numerous, efficient, and mobile. They can detect chemical signals from food sources from significant distances across the featureless trench floor. They arrive in large numbers when food concentrates, process it quickly, and disperse. Their bodies become the primary food source for the snailfish above them. The snailfish is the endpoint of the vertebrate food chain in the trench. It feeds almost exclusively on amphipods and small crustaceans. It has evolved a body optimized for this environment, soft, pressure-tolerant, energy-efficient. It moves slowly, conserving energy in a system where calories are scarce. Every step of this food web is built on efficiency. In a system with limited energy input, no organism can afford waste. The metabolisms of Hadal creatures are slow by surface standards. Their growth rates are slow. Their reproduction rates are slow. A snailfish may live for years before it is fully grown. A deep-sea clam at a seep community may live for decades. This slowness is not failure, it is precision. Every calorie that enters the system gets used. The Hadal food web runs lean because it must. What the 2025 research added to this picture was the role of microbes at each step. Scientists found that microbial communities are not just at the base of the food chain. They are embedded in the bodies of animals throughout the chain, processing chemicals that their hosts cannot handle alone. It is a web of webs, and the more you look at it, the more clearly it suggests something about life on other worlds. On land, biology follows a set of assumptions so consistent that most biologists have never had reason to question them. Bones are rigid because they need to support weight. Cell membranes are semi-permeable because they need to control what enters and exits the cell. Proteins fold into stable shapes because those shapes determine their function. Gas-filled cavities allow organisms to control buoyancy or breathe. Every one of these assumptions breaks down at 6 miles below the surface. Rigid bones crack under extreme pressure. At the bottom of the Mariana Trench, the snailfish survives because its bones never hardened. Its cartilage-based skeleton bends instead of shattering. What would be a developmental defect on land is a survival advantage in the deep. Gas-filled cavities implode. Any creature with lungs, a swim bladder, or a gas-filled sinus would be destroyed before reaching the Hadal zone. Every animal that lives there has solved this by eliminating gas spaces entirely. Their bodies are fluid-filled, water-dense, built to equalize pressure rather than resist it. Protein folding becomes unreliable. Pressure distorts the molecular environment that proteins need to maintain their shape. The solution evolved in the deep ocean, using molecules like trimethylamine N-oxide to stabilize water structure around proteins, is a category of adaptation that surface biology simply does not need. Cell membranes solidify. The fats that make up cell membranes change behavior under pressure. At surface conditions, most biological fats create membranes with the right balance of rigidity and flexibility. At extreme depth, those same fats would stiffen too much, making membranes brittle. Deep-sea organisms evolved to use different fats, specifically polyunsaturated fatty acids, that stay fluid at high pressure. What emerges from this list is a picture of biology that is completely contingent on its environment. Every rule of biology that surface life follows is not a universal law. It is a local solution to a local set of physical conditions. Change the conditions dramatically enough, and life changes the rules it follows. The Hadal zone is a natural laboratory for finding out which rules of biology are actually rules, and which ones are just habits of surface life. The implications of this are not abstract. They directly affect how scientists think about life elsewhere in the universe. Because the same question that the Mariana Trench asks, how adaptable is life to extreme conditions, is exactly the question that determines whether life can exist on other worlds. And the answer the trench keeps giving is the same every time, more adaptable than we thought. Methane is one of the most potent greenhouse gases on Earth. Molecule for molecule, it traps roughly 80 times more heat in the atmosphere than carbon dioxide over a 20-year period. It is produced naturally by the breakdown of organic material in sediment, by geological processes in the Earth's crust, and by microbial activity in a wide range of environments. The trench floor leaks methane. Tectonic activity drives it up through cracks in the rock. Cold seeps release it slowly and steadily into the water above. If that methane made it all the way to the surface and into the atmosphere, it would represent a meaningful contribution to the planet's greenhouse gas budget. It largely does not make it, and the reason is microbial. Scientists working with samples from the 2025 Fenduji mission found methane-consuming bacteria in the sediment throughout the trench floor at extraordinary densities. These bacteria, known as methanotrophs, consume methane as their primary energy source, converting it into carbon dioxide and water. Carbon dioxide is also a greenhouse gas, but a significantly less potent one. The methane-consuming bacteria in the Mariana Trench are performing a chemical conversion at depth that prevents a fraction of the Earth's naturally produced methane from ever reaching the atmosphere. They are functioning in effect as a biological filter built into the floor of the ocean. The total scale of this process across all deep-sea trenches worldwide is still being calculated. Scientists are cautious about claiming it represents a large-scale climate regulator because the data is not yet comprehensive enough. But the mechanism is real. The bacteria are there, they are consuming methane, and there are trench systems similar to the Mariana Trench distributed across every ocean basin on Earth. What is most striking is that this function was entirely unknown before systematic sampling of the Hadal Zone began. The trench was understood as a geological feature, an extreme biological environment, a collector of marine snow. Nobody had included it in global models of methane cycling. The 2025 data forced a rethink. The deep ocean is not a passive recipient of what falls from above. It is an active participant in the chemistry of the entire planet. The Hadal Zone is performing planetary maintenance, and until recently, it was doing so entirely without our knowledge. The floor of the trench is not as still as it looks. In 2015, scientists from the National Oceanic and Atmospheric Administration lowered a hydrophone into Challenger Deep, the deepest point of the Mariana Trench. They expected the recording to be quiet. 6 miles of water above, no shipping lanes directly overhead, no obvious sources of sound. The expectation was near silence, the kind of acoustic emptiness you might associate with a place this remote. What they got was a busy, complicated soundscape. The baleen whale calls came through clearly. Long, low-frequency vocalizations that had travelled through the Pacific's deep sound channel, from whales that could have been hundreds of miles away. The depth of the trench and the properties of water at that pressure make it an exceptionally efficient receiver for low-frequency sounds. Ship propellers were audible, not nearby ships. The recordings captured the distant rumble of commercial vessels crossing the Pacific far above and far away. The sound had travelled through the ocean and into the trench, arriving at the hydrophone as a low, steady throb. Earthquakes registered. The Mariana region is one of the most seismically active areas on the planet. The hydrophone recorded multiple seismic events during its deployment, each one registering as a sharp, reverberant burst of acoustic energy in the water, and at one point, a Category 5 typhoon passed over the area at the surface. The winds and waves generated sound that penetrated 6 miles of water and was detected at the bottom of the trench. The lesson from all of this is counterintuitive. The most isolated place on Earth is acoustically connected to the rest of the planet. Sound from typhoons, from ships, from whales, from tectonic events. All of it reaches the bottom of the Mariana Trench. The creatures living there exist in a sonic environment shaped by events happening at the surface and thousands of miles away. For animals that evolved in the absence of light and depend on detecting vibration and chemical signals to navigate their world, this constant acoustic environment is their version of the news. Every sound that travels down tells them something about what is happening above. They have been listening to the planet for millions of years. What the sediment under them records is something entirely different. The floor of the Mariana Trench is not still. From the surface, or from the porthole of a submersible, the sediment looks motionless. A flat, grey-brown plain of fine particles barely disturbed, apparently unchanged for centuries. But the walls of the trench are steep, and steep walls plus loose sediment plus the occasional earthquake creates a dynamic very different from what the visual suggests. The Mariana Trench experiences sediment avalanches. When seismic activity shakes the trench walls, loose material, sediment, organic particles, small rocks, slides downward along the steep slopes and accumulates at the bottom. These underwater avalanches, called turbidity currents, can move quickly and can redistribute material across significant areas of the trench floor. The result is a floor that shifts. Sediment layers are not always laid down in perfect horizontal sequence. They get mixed, overturned, and redeposited. Dating the sediment in the Mariana Trench is technically difficult precisely because of this mixing. Scientists have also found that the depth of the trench itself may not be perfectly constant. The subducting Pacific plate is still moving, the floor is still being pulled down. Some measurements suggest the geometry of the trench changes over geological timescales. with the deepest point shifting slightly as the plates continue their slow grind. The fine sediment covering the floor is a record of everything that has sunk from above over millions of years. Compressed into layers, it contains dead microbes, mineral particles, traces of ancient plankton blooms, and, in the most recent layers, microplastic fibers. Reading those layers is like reading a history of the ocean above. The sediment also contains living microbes in extraordinary densities. Scientists taking core samples have found active microbial communities, not just at the surface of the sediment but tens of feet deep into it. These microbes are processing organic material that has been buried for thousands of years, working through it at rates that are almost unimaginably slow by surface standards. At 6 miles down, time works differently. A process that happens in days at the surface may take decades in the sediment of the Hadal zone. A microbe living in the deep sediment may go through 1 cell division per year rather than dozens per day. And yet these slow, buried microbes are processing material, cycling nutrients, and releasing compounds back into the water column above them in ways that affect ocean chemistry globally. The bottom of the ocean is slow, it is moving, it is alive, and what the temperature of that living floor does to the creatures surviving on it overturns one more assumption most people carry. The water at the bottom of the Mariana Trench sits at roughly 34 degrees Fahrenheit, just above freezing. Most people's intuition is that cold means slow, cold means less energy, cold means sparse, struggling life barely holding on. The bottom of the Mariana Trench is cold, and it is densely populated by the standards of deep ocean environments. The relationship between temperature and biological activity is real at the surface. Tropical reefs are more biologically active than polar seas. Warm water speeds up metabolic reactions, cold slows them down. But in the Hadal zone, the pressure partially compensates for the cold. High pressure alters the behavior of molecules in ways that can actually increase the rate of some chemical reactions, even at low temperatures. The two forces, cold-slowing reactions, pressure-altering reaction dynamics, interact in ways that surface biology does not prepare you for. Deep-sea microbes adapted to cold and high pressure are called piecophiles, pressure lovers. Many of them grow better at high pressure than at low pressure. Bring them to the surface and they stop functioning. They're adapted so specifically to their extreme environment that normal surface conditions are as hostile to them as the deep is to surface life. The cold also has one significant advantage in the deep ocean. It slows the decay of organic material. A piece of organic matter that would decompose in days at a warm surface temperature can persist for much longer at near-freezing temperatures under high pressure. This means the food available to deep-sea creatures, however sparse it is, remains available longer. The cold is not only a challenge, it is also a kind of preservation system for the scarce resources the Hadal food web depends on. The snailfish navigating the dark floor in near-freezing water at crushing pressure is not suffering. Its biochemistry is calibrated to these conditions. Its enzymes operate at peak efficiency in the cold. Its cell membranes are built for the temperature. It is as comfortable in its world as a reef fish is on a sunlit coral. Understanding this changes how scientists think about the hard boundaries of life. There are no hard boundaries. There are only environments life has not had enough time to adapt to yet. The adaptation of Hadal creatures to extreme pressure is so complete that reversing the pressure kills them. This is one of the practical problems that makes studying the biology of the Hadal zone so difficult. To get a live specimen from the bottom of the Mariana Trench, you have to bring it up through 6 miles of ocean, while the pressure drops from 16,000 pounds per square inch to 14 pounds per square inch. That transition happens over hours, but it is still a rapid pressure change relative to the evolutionary timescales these animals adapted on. For the snailfish, the consequences are fatal. During ascent, dissolved gases in the tissues may behave differently than at depth. The gelatinous body, built for high pressure, does not handle the change well. every snailfish brought up from the Hadal zone has died before reaching the surface, or died shortly after. This means that essentially everything science knows about the live biology of the snailfish comes from footage captured at depth, from dead specimens examined immediately after retrieval, or from analysis of frozen or preserved tissue. Scientists have never studied a living snailfish in a laboratory. For microbes, the situation is somewhat better. Microbes preserved under pressure in sealed containers can be kept alive during ascent if the transition is managed carefully. Some research facilities have built pressurized vessels specifically for culturing deep-sea microbes at their native pressure. These are expensive and technically demanding, but they allow scientists to study deep-sea microbial metabolism under conditions that actually match where the organisms live. The organisms that survive the ascent most reliably are the ones with the most flexible biology, microbes with larger genomes that can adjust to changing conditions, or invertebrates that live across a wider depth range and are less precisely calibrated to one specific pressure. The organisms that die on ascent are the ones most specifically adapted to the extreme. Their precision becomes a vulnerability the moment the environment changes. This is a biological truth that applies everywhere, not just the deep ocean. Extreme specialization is a survival advantage in a stable environment and a fatal liability in a changing one. And what the organisms of the Mariana Trench have been teaching scientists about life in extreme environments reaches much further than the Pacific Ocean. It reaches 63 million miles away, to a moon covered in ice. Europa is one of Jupiter's moons. Its surface is a cracked sheet of ice. Below that ice, scientists believe, sits a global ocean of liquid water, roughly twice the volume of all of Earth's oceans combined. That ocean sits in total darkness. It has no access to sunlight. It sits under an ice shell dozens of miles thick. It is in contact with a rocky seafloor. Scientists believe that tectonic activity at the base of that ocean may produce chemical vents, places where mineral-rich water seeps out of the rock, powered by the gravitational forces Jupiter exerts on Europa's interior. This description is nearly identical to the conditions found at cold seeps and hydrothermal environments in the Mariana Trench. Dark, pressurized, cold, chemical energy from rock, no sunlight. If life can form a thriving ecosystem at the bottom of the Mariana Trench under those exact conditions, the question of whether life could exist on Europa stops being speculative and becomes testable. NASA astrobiologist Kevin Hand has written extensively about this connection. The research program he advocates starts with Earth's deep ocean, specifically environments like the Mariana Trench, and uses what we learn there to build models of what life on Europa might look like, where it might be, and how to detect it. The chemosynthetic bacteria at the base of the Mariana Trench's seep communities are the closest analogues we have to what Europan life might be. Single-celled organisms deriving energy from chemical reactions between water and rock. No sunlight required. No photosynthesis. Just chemistry and pressure and time. The tube worms and clams of the deep-sea seep show that multicellular life can evolve on top of that microbial foundation, even in total darkness and extreme pressure. This makes the possibility of complex organisms in Europa's ocean not just microbes, but something larger, harder to dismiss than it was before the Mariana Trench's seep communities were fully documented. The 2025 discovery of the deepest chemosynthetic communities ever found strengthened this case further. Every time science discovers life surviving in a more extreme environment than previously documented, the case for life on Europa, on Enceladus, on any world with liquid water and chemical energy gets proportionally stronger. The Mariana Trench is not just an extreme corner of our own planet, it is a reference point for the universe. Before 1977, the definition of a habitable zone had one component: distance from a star. A planet was considered potentially habitable if it sat at the right distance from its star to have liquid water on its surface. Too close, and the water evaporated. Too far, and it froze. The Goldilocks zone. the narrow band around each star where conditions were just right. That definition assumed one thing: life needs sunlight. Because life on Earth needs sunlight. Because every ecosystem scientist had ever studied ran on photosynthesis, which runs on sunlight. The assumption was so embedded in biological thinking that it was not even recognized as an assumption. It just seemed like a fact. In 1977, scientists diving in the Alvin submersible near the Galapagos Islands found hydrothermal vents on the ocean floor. Dense communities of tube worms, clams, bacteria and fish were thriving around superheated water venting from the seafloor. No sunlight, no photosynthesis. The entire ecosystem ran on chemosynthesis. This cracked the definition. Life does not need sunlight. Life needs energy, and energy can come from the inside of a planet. Every subsequent discovery in the deep ocean has widened that crack further. The cold seep communities, the chemosynthetic tube worm colonies at 6 miles down, the methane-consuming bacteria processing greenhouse gases in trench sediment, 7,000 microbial species in the darkest part of the ocean, each one running its metabolism on a different chemical trick. The habitable zone around a star is still a meaningful concept for surface life, but for life that runs on chemosynthesis, like everything in the Hadal zone, the location relative to a star is irrelevant. What matters is liquid water, a source of chemical energy, and enough time for evolution to work. Liquid water and chemical energy exist inside planets and moons all across the solar system. Europa. Enceladus, Saturn's moon, which actively vents water into space from its subsurface ocean. Ganymede, possibly Titan, possibly ocean worlds around other stars entirely. The Mariana Trench did not just expand our understanding of life on Earth, it expanded the number of places in the universe where life could exist by removing the requirement that life be near a star. That is one of the largest scientific revisions of the last century, and it came from 6 miles below the surface of the Pacific Ocean, from a place most humans will never see. But what remains unseen even there is the most humbling part of this story. Of all the facts in this story, one lands harder than any other. Scientists have explored less than 20% of the Earth's total ocean floor in any meaningful detail. Less than 20%. The ocean covers over 70% of the planet's surface. The deep ocean, below 1.5 miles, represents the largest continuous living space on Earth, and we have barely looked at it. The Mariana Trench specifically, the single most studied Hadal environment on the planet, has been visited by manned submersibles fewer than 24 times in recorded history. The combined amount of time humans have spent physically present at the bottom of Challenger Deep is measured in hours, not days. Every single expedition has found something that was not expected. Every sample brought up has contained organisms or molecules that required science to revise something it thought it knew. The pattern is consistent and clear. The more carefully we look, the more we find, and the more what we find differs from what we predicted. 7,000 microbial species in one trench, a new species named after the plastic inside its body. Chemosynthetic colonies deeper than anyone thought possible. Methane-consuming bacteria performing planetary chemistry nobody had included in global models. Pressure-adapted genes with potential pharmaceutical applications. A complete alternative food web running on chemical energy from the Earth's interior. All of this from less than 100 cumulative hours of human presence at the bottom of one trench, in one ocean, on one planet. The Mariana Trench stretches 1,580 miles across the Pacific. Manned missions have visited a tiny fraction of that distance. The trenches running under the Atlantic, the Indian Ocean, the Southern Ocean. Many have never been visited at all. We have mapped the surface of Mars with greater resolution than we have mapped the floor of our own ocean. We have a more detailed picture of the far side of the Moon than of the deep Pacific. The most extreme environment on our own planet remains in most of its extent completely unknown. What lives at the bottom of the Java Trench, the Puerto Rico Trench, the deepest points of the Southern Ocean? Science has fragments and guesses. The systematic biological surveys that revealed the Mariana Trench's secrets have barely started in these other systems. The deep ocean is not just an unexplored place, it is an unexplored world sitting under 6 miles of water on a planet that 7 billion people call home. It ran its own separate experiment in biology for hundreds of millions of years. It built food webs without sunlight, pressure-adapted molecules without engineers, chemical systems that clean the planet's atmosphere without anyone asking it to. And the most important thing it has done, the thing that matters most for how we understand life in the universe, is prove, over and over, that life finds a way. Not the way we expected, not in the places we looked first, not following the rules we assumed were universal. Just life. Stubborn, creative, endlessly adaptable life. 6 miles down in the dark, eating poison, in the cold, under pressure that would crush everything we have ever built. Still there, still going.